Well, I'm here at the John Innes Centre near Norwich at their research plots, and I'm here to see what could be one of the most remarkable wheats we've ever seen in this country. This is Watkins 579. To tell me what's remarkable about this wheat, I've come here to see Maria.
Hello, I'm Maria. I'm a researcher at the John Innes Centre, and for the last 5 years I have been exploring traits that have been not evaluated before in wheat. So for that I take advantage of our collection of wheat from all over the world. It's called the Watkins collection. And what is interesting about this particular wheat in this plot is that it has a demonstrated capacity to decrease the transformation of ammonium into nitrate in the soil. And that is just by putting some compounds, but very specialized compounds in the soil.
Well, this is something that a wheat does naturally. When you talk about these compounds, they're just compounds that the wheat produces naturally, is that right?
Absolutely. You have to see that these varieties are originally from Iran, from all the Persia area, and it's kind of an adaptive trait. So they evolved to put these compounds in the soil so they could make the most of soils where nitrogen is not particularly abundant.
Okay, so just explain why that's useful. So you talk about, so we put nitrogen on the soil, ammonium nitrate and the urea. What is it that these wheats are then doing to that nitrogen?
They are slowing down the activity of the bacteria that does that transformation. So when we put our urea or our ammonium, these bacteria will have a lot of food to process, and then they will proliferate and they will transform the ammonium into nitrate really, really quickly. That's a problem. So it's a good thing in a way because the plants want both ammonium and nitrate, but it's bad because it's very soluble, so the plant doesn't really have time to absorb everything. And because it's very soluble, before the plant can take it, it's already run off or even volatilized and contributes to nitrous oxide.
So what we're talking about here is an ability to slow down that process. So rather than, because we all know that nitrogen is not very efficient. It's about sort of 50–60% efficient, I think, in most fields, isn't it? Yeah. And so that means that a lot of the nitrogen that we're applying as farmers is just leaching out into the soil
average,
water.
it's 50% is completely lost.
So just remind me again that so these wheats have got a compound. They produce a compound. Do we actually know much about this compound?
We have identified some similar compounds in sorghum, in rice, and in some wild grasses, but not yet. We know it's there, but we haven't identified what is yet.
And this wheat does it. Are there any other commercial wheats that do this?
We haven't found that in commercial wheat yet, so that it's remarkable. So we do several assays. So we look at the microbes directly. We just take the soil from the roots, look at the microbiome composition. We also do little assays in the lab. And we see really that not just that there is a lower transformation rate, there is also much more nitrogen going into the seed.
Okay, well, that's the potential here, that we've got a nitrogen, a wheat that moderates its own nitrogen in the soil. Is that right? So that means less going down into the soil water or into the air, and more being taken up by the wheat because it's controlling the amount that it needs, really, isn't it?
To quite a good extent, actually.
Now, we're making more of this, aren't we? So we're multiplying it up, and there's a plot here at the Baldborough research station somewhere that we're going to be harvesting shortly.
Very soon, yes.
And then we're going to be distributing that to our farmers within the Truth Project next year, isn't it? This is one of the aspects that we're looking at. So we're doing some on-farm trials with 20 of our root rangers who are going to be using some of this wheat. They're going to be growing this on their farm next to some of their farm standard wheat. And we're going to be looking quite closely at the roots, aren't we? So tell us about the sampling that we're doing on the roots.
Once the fertilizer is applied, there is going to be this kind of uprise of nitrifying bacteria and other organisms. So it's at this moment. So we have a window of like between 5–7 days after the application of the fertilizer when we have to go and act, and we take some samples. And then we look at the microbial composition. So we are doing metagenomics, which is the best approach we have at the minute available. And we combine that with some measurements in the lab of the actual chemical transformation. So we can prove that it's not only that there is a suppression or a control of the community, that is actually a suppression of the action.
Right. By comparing the roots with this wheat with a normal wheat, then we'll get a good idea of just how much microbial action is going on, and whether these really do have this ability to moderate their own nitrogen. Is that right?
That's correct.
Fantastic. Well, look, Maria, I'm really looking forward to this trial. Looking forward to harvest, to getting the plots harvested, and then we're going to be multiplying them for another year before we give them to our farmers. Each of our farmers is going to be growing about 1 hectare of these plots. So that's going to be a lot of wheat. There's going to be a lot of data coming in. It's going to be a really exciting project. Thank you very much for showing us this wheat.
My pleasure.